This is a component of the bio367 lecture on prokaryotic genetics, and it addresses the processes of transduction. Transduction is one of the three methods whereby prokaryotic cells can experience the movement of DNA to an unrelated neighbor, the process of horizontal gene transfer, or HGT. In transduction, we have the transfer of DNA from one cell to another via a bacteriophage, or virus that can infect those cells. Transduction can be either generalized or specialized. It occurs in many bacteria and involves many different types of phage, but not all. Okay, so in other words, there are some bacteria that just simply do not experience the process of transduction, and there are others that do. Many of the organisms found in marine environments we know can be transduced. It appears to be a very important mechanism of horizontal gene transfer in nature, particularly in marine environments. Earlier in class, we looked at bacteriophage, and we looked at their two types of life cycles. We talked about bacteriophage that use the virulent or lytic lifestyle only, and we talked about those that are temperate. In other words, they can switch back and forth between lysogeny and lytic lifestyles. Generalized transduction involves only those phage that are acting in a virulent or lytic manner, okay? Which essentially can be all phage. It's just that in the temperate phage, they only do that once in a while, but while they're doing lysis, then generalized transduction can occur. In generalized transduction, we can only transfer bacterial DNA, and the transfer of DNA occurs in essentially unspecialized or generic chunks. Okay, so what will happen is the phage will adsorb or bind to the host cell. It'll inject its DNA, and then havoc will occur in the cell. So the cell machinery will be forced to replicate the viral protein and the viral genome, and you will get so many new phage constructed that they will eventually pressurize the cell to the point where it bursts, and out will go the new phage. And the cell is dead, the cell is lysed. Okay? What can happen in this sort of genetic free-for-all is that occasionally, as the phage proteins spontaneously assemble, because that's a very spontaneous process, they can encapsulate not viral DNA, which is shown here in red, but a little bit of the host DNA, because the host DNA is being degraded as all of this happens. So this phage right here is not quite right from the point of view of how the virus should be constructed. In that phage's nucleocapsid, you've got host DNA instead of viral DNA. So when this organism, well, it's not an organism, when this phage goes off, it will be able to adsorb to a new host cell, and it will inject that genome into the new host cell. But once that DNA gets in there, it's not going to act as viral DNA, so you're not going to have an infection. Instead, what can happen is that green DNA can find regions of homology in the new host cell, and recombination may occur, which can alter, genetically change the new host cell in the future. Only if recombination occurs, however. So it's called generalized transduction because it's really any piece of DNA, like any of this chromosome could have ended up in there, right? So you can't predict which piece of DNA will end up in there. This is a very low efficiency process, okay? It depends on a random error, and then it depends on homology and recombination occurring, so this doesn't happen at a very high rate. Nonetheless, it does happen in nature, and it is a mechanism of introducing genetic diversity. Now, just to review, let's consider our lysogenic or temperate phage again. So this is a slightly different cartoon than I showed you earlier, but bear with me, okay? So with a lysogenic phage, they can spend time in the lytic cycle, and they do that whenever they need to move to a new host, but they can also spend quite some time in the lysogenic or temperate cycle. So here we have a bacteriophage that's attached to a host cell, and it's injected its DNA, okay? And that DNA is found here. And some of the time, that DNA can actually integrate into the host cell chromosome. And when that happens, we refer to the virus now as a prophage, okay? Because it's a little bit different. And over binary fission for sometimes many, many generations, the prophage will persist and it will be replicated along with the rest of the host cell chromosome. At some point in time, however, certain environmental circumstances can trigger the excision of the viral DNA from the host cell chromosome, and it will return to its viral DNA state, and then that viral DNA will be expressed, and you'll enter the lytic cycle. Okay, you'll get new viral DNA, you'll get new viral protein, new phages will form, they'll pressurize the cell until it bursts, out they go, and then they can infect new hosts. Now, the process of specialized transduction depends on this process occurring. So specialized transduction can only occur when you have infection by lysogenic / also known as temperate phage. All right, so here's specialized transduction, and it's shown over here on the right, okay? So picking up from kind of where we left off, although the cartoon's a bit different, here we have a lysogenized cell, okay? That's the phage DNA. It's in its prophage form, all right? So the virus got in and its DNA integrated into the host cell chromosome, okay? And time went by, and then something in the environment triggered the phage DNA and told it to come out again and move over into the lytic type of life cycle. Normally, that would happen as shown here on the left, which I'm not going to go through. But once in a while, what can happen is when the phage or viral DNA excises, it can take a portion of the host DNA with it, and that's shown here in red. Okay, so that red DNA is host cell DNA, it's bacterial DNA. It's excised and it's there and it's being replicated, okay, and then new phages are being assembled and out they burst, and every single one of these hundreds of phages now is able to go off and infect a new host cell, and it will inject that DNA within it, and that DNA is carrying not all viral DNA, but some of the host cell DNA. And that means that there's the possibility for recombination to occur and genetic differences. This is a very efficient process. It was first discovered in E. coli, and it's infecting bacteriophage called lambdaphage. It's very efficient, and by that I mean it happens at a pretty high rate because every single one of these phages has that host cell DNA in it, not just one from the batch. All of them have it. So when they go off, there's a really good likelihood that that host cell DNA is going to be introduced into the new recipient cell. Okay. It's also called specialized because only those genes that are located next to the phage DNA, back up here, only those genes can be transferred. So it's pretty specific which piece of DNA can be moved. It has to be a piece of DNA that is immediately adjacent to the viral DNA, or it can't be transported out into new host cells in this manner. Okay, a few study guide questions. You should know sort of the basics of bacteriophage and their life cycle, so you should know the difference between bacteriophage that are entirely lytic and those that can have lysogenic or temperate lifestyles. You should also know which participates in general transduction and which participates in specialized. Okay, generalized transduction happens whenever you've got infection that results in lysis. So, in fact, either type of phage can do generalized transduction or general transduction. Specialized transduction only happens in those phage that can participate in making a prophage. That's only those that are the so-called temperate or lysogenic phage. Question 3 is, do bacteriophage that conduct lysogenic lifestyles use specialized or general transduction? Here the answer is both. but we tend to study them more from the point of view of specialized transduction. And then, do both lytic and lysogenic bacteriophage lyse cells? And the answer there is yes. It's just that lytic phage will lyse them immediately, lysogenic ones just from time to time when the environment triggers that aspect of their so-called life cycle. And that concludes this part of the lecture.